All right. Thank you, everyone, for coming to this presentation in the Science Circle Saturday series. I am your presenter today, Stephen Gasior, known in Second Life as Stephen Zootfly. I am a researcher in a completely different industry than aging, uh, but this is one of my kind of favorite topics to come back to just because it's another one of these particular fields that has a lot of exciting things going on, a lot of impactful things for people, and I think is also a place where, in particular, there can be a lot of misinformation given all the excitement that it has. And Syzygy says it's a subject that never grows old, and hopefully one day that'll be unlike us, right? Again, we can actually have a little conversation about whether that's a good or bad thing. But let's go ahead and get started because I think what's really exciting right now, and you might have seen some of the headlines, is that there is a reverse aging trial. And when you put it in those few words, that actually creates, on the one hand, in the short term, a very incomplete and over descriptive picture of what's going to be happening. But I think it is something that, depending on how it turns out, the underlying idea of why it's a clinical trial and why it might work can actually be quite impactful over the long term. So, eternal youth and immortality, two things that have been a part of the human condition since the beginning, at least as far as we can tell of literature, maybe even in the spoken mythology word. What I have up here is a selection of artwork. The Alchemist up on the top left, a depiction of people interested in finding the Philosopher's Stone in order to make the elixir of life. Top right hand illustration or painting of Ponce de Leon interacting with Native Americans. Again, he was on a quest to discover the fountain of youth. And again, something that is a mythology that you see repeated, the quest for the fountain of youth in many, many stories. The lower left is an interesting one. This is not just a Western phenomenon. That is some sort of bunny that lives on the moon that is currently stirring the elixir of life from Chinese mythology. And of course, this is something that does not get old, where, again, the most recent movie I can recall myself having seen where this is the theme is The Jungle Cruise, based on a theme ride at Disney. But basically, again, a quest for a tree that imparts can healing powers primarily, but also youth, vigor, and immortality. And so I start off with this, and I'll post the web page for this one in local chat. Is this has been around forever, right? And that really what we have is two parts of the human condition, which is one, that we do grow old. This is inevitable. We do age. And the other thing is that because we have some expanded, amazing mental capacity, we both envision and can predict that this will happen and thus fear it and wish it to not happen. And so we will, in many cases, try to struggle against the dying of the light, as they might say, in order to have this not happen. But I do want to point out one thing here, and that is in this talk, one thing I want to be very specifically important to talk about is that when we talk about reverse aging, none of these aging remedies, shall we call them, or interventions are something that can actually stop a different clock, which is the mortality clock. And so aging, I want to point out, is something separate than our inevitable mortality due to the shortening of telomeres. And so I won't get into the details of that, but one thing that's really important to keep in mind here is that a lot of this reverse aging is really just about returning to healthfulness. And healthfulness is of value regardless of your age. And it's about how we enjoy what life has to offer. So what I have here is a small selection of things that we associate with aging. Again, if I were to ask you what is aging, actually your answer might just be growing old. And in fact, that's not, starting from when we're born, we're aging, right? I mean, we can conceptualize and we see these changes that occur as we age. But when we think about old age, gray hair. Gray hair is actually the most highly penetrant phenotype of aging, although in many ways, one of the extremely least consequential in terms of our health. What we also have on the top right is an increased association with neurological impairments. We definitely, even as a healthy person, we can slow down. Our brains don't operate as fast and as agile as they used to. But there are several neurological pathologies. And what I have here is a picture of amyloid plaques that are associated with Alzheimer's. There's Parkinson's, there's Huntington's, there's frontotemporal dementia. So again, these are diseases you don't phenotypically see, although you can in some ways measure them at an earlier age, but they're definitely something of the over 50 to 60 years of our lives. And then hormone levels. Again, this is something you think about that, again, as you get older, you start saying, wow, I don't feel like I had the ability to lift weights or to recover from an injury or just the vigor and the energy that I used to. And so what I have here is a graph of average testosterone levels in males over time as one example of that. And again, there's definitely a feeling that you have when you're getting older. Again, some of these hormones impact things like skin elasticity, how well your diet works, how well your immune system is working. Women, as Sis points out, testosterone is important in women too. That's still a part of what their hormones are. But women also go through a very interesting case with menopause, right? That's a distinct happening that occurs in older women. Uh, Probably the one that we want to think about the most, and I think this is where we want to philosophically get to a different point about thinking about it, is the idea of disease susceptibility. So what I have on the lower right is the rates of hospitalization for COVID-19 associated with different decades of age. And again, this is from March of 2020. One thing you can distinctly see here is that the severity of the disease as indexed by having to go to the hospital uh, is much, much higher as you get to be over 50 years old. And in fact, if we were to look at mortality rates, long-term effects, we would see that as well. And so there's something definitely going on. So aging, although we kind of know it when we see it, we don't necessarily have an exact way of quantifying and thinking about it. But we do have lots of epidemiological statistics, of course. And so here's another one with cancer risk. We know that cancer is something that has to do with genes going wrong, chromosomes going awry, misregulated growth, other things happening with cells that make them malignant and eventually create some sort of lethal effect as they overgrow and overtake our tissues. And that, again, you can see the risk of cancer dramatically increases really over the age of 50 compared to when you're under 30. And so now what's interesting about cancer is that we actually have some of the things that are associated with aging as well. And one thing that people have done starting in the early 2000s is to think about aging as something that's quantifiable, something that in fact has aspects of disease, just like cancer has aspects of disease. There are aspects of aging, the things that are happening to our cells that are aspects of aging that we can quantify as if it were something like a disease. And that's what we have in the next slide. Again, this is a more recent iteration of this idea. This has been around since 2013, but something called the hallmarks of aging. Now, again, these are not necessarily the causes of aging, although some of them probably certainly are. But these are things that are associated with aging that we can measure, that we can quantify at a cellular level, and see how it impacts the overall health and well-being of the whole organism. And so I'm going to list just a few here that things you might have heard about are telomeres get shorter. There's things known as epigenetic uh, alterations. Protein functioning and protein recycling may not work as well. Something called genome instability. Senescence is a term that you might have heard about. And so uh, stem cell exhaustion, that you need to have stem cells in order to replenish like your skin or other cells in the body. And so there's a lot of things that we know that we can see that we associate with aging. There are aging diseases where these are accelerated aspects of aging diseases and whatnot. And so let's talk about some of these after we have just a quick conversation about why aging, right? And so there was a quote in local chat from put there by somebody, by Phil, talking about Steve Jobs, that Steve Jobs has described death as, quote, very likely the single best invention of life, end quote. Right? Where would evolution be? Where would we be if people didn't die? Right? The fact that we are beings of a transient state means that there can be another generation. And yeah, a term put in there, planned obsolescence. Now, from an evolutionary biology point of view, that's not necessarily the case, right? There's not necessarily a program in which our cells say, oh, yeah, we are going to do these things that ultimately do promote death. Although that is still out there. There are people who argue that that may be the case. It's hard to say. One thing that I think is, in my point of view, for all cellular organisms or anything that you construct, is that there is this inevitability of thermodynamics and entropy that can cause decay, right? Now, we as biological organisms overcome entropy in a certain sense, in that we can use energy to build structures, to build metabolism, to replenish and rejuvenate various cells and whatnot. And so, but again, obviously, I don't want to argue with the physicists. The overall entropy does increase because we're using energy to make that happen. But there are these programs in life, right? We start out small, we're multicellular. We go through adolescence. We go through growth phases. We go through reproductive cycles. And then, uh, like many organisms, once we've had our reproductive time, is there a lot of purpose to being around? And so there's not an evolutionary drive necessarily to maintain our ability to um, to live longer. Now, there are some countervailing forces there. When you think about parental care, when you think about grandparental care, when you think about grandparental care, when that helps the survival of organisms, there could be a selection for those things. And in fact, one reason that we do have potentially such long lives is that we are engaged in parental care. So there has to be some degree of our genes helping us understand that. And yeah, that does help the survival of the organism. And so again, it depends on the societal structure of many organisms, but you know, aging is immortality is also not hardwired into every organism. There's actually a Medusa organism that actually rejuvenates its entire life plan and goes through cycles of living, so that in fact, it's in essence, it's essentially immortal. It deprograms all of its cells and goes on through another life cycle. So this is biology. Mother Nature's a mad scientist, a famous philosopher once said. And so what I really want to point out is, well, we don't have a solid evolutionary theory on why this happens, is that at the cellular level, we can think about the causes of this, whether it's programmed or not programmed, and say, are there places in which we can intervene, and not necessarily to become immortal, but at least to give ourselves healthier lives as we go there. All right, so I'm going to focus in on some of that chemistry, right? There's a little bit of chemistry we are familiar with in terms of the aging process, in terms of hallmarks of aging and how cells work. And then that really understands a lot about how cells work. And this is something called methylation. And so methylation is a tag. It's a little bit of chemistry that goes onto our DNA. And when that happens, it also recruits the protein machinery that helps compact our chromosomes. Again, one thing to keep in mind is that our genome, if you were to take our DNA and stretch it out from one single cell, it would actually be 2 meters long. And so that doesn't fit in a cell. And so what happens is every single cell that we have compacts it down in a way that makes it easier to keep inside the cell. And then, but when you package it down so tight, you also have to leave it open and accessible if you want to have genes be on or off. And so without going into a lot of detail on this, just keep in mind that methylation status tells us a lot about whether parts of our chromosomes are on and that the vast majority are off. And you can only be a working gene if your chromosome status is to be open. And a tagline points out something here, just to come back to that philosophical thought. There are now some interesting fictional accounts of what happens with immortality. One that I have watched and one that I might recommend is a show called Altered Carbon, in which alien technology allows people to keep re-downloading their memories into a new body. And of course, they have cloning technology, so you can basically clone your body over and over. Altered Carbon. Altered Carbon. Again, it's a show for adults. Let me be very clear here. It is a show for adults. And it's got a lot of graphic stuff in it too, but it's an interesting case. What happens to society when people are, from a legal standpoint, essentially immortal when it comes to their belongings? So, but methylation is this key thing. And in fact, methylation has actually been developed in a way to something called methylation clocks. Just a quick question. Has anybody here, yourself, or know of anybody who's had their methylation clock measured? This is something you can now do with a mail-in order service. You take a cheek swab, you send it to somebody, and they tell you what your methylation age is, in essence. And so what has been done with enough methylation data, analyses, and longitudinal data is that people like Horvath have developed these methylation clocks that are actually strong predictors of mortality risk. All right, so these aren't necessarily a clock in terms of age, but what you can do is associate them with, in a sense, an aspect of aging, which is how much longer do you have to live? And then you can normalize that against other people. Like a 50-year-old could have a methylation clock that says they're more like they're 43, or a methylation clock that says they're more like 70. What I have here in terms of how these get developed and how they think about them is from a more recent paper, 2019, by Horvath, where they talk about the generation of these methylation clocks, biomarkers, and then these regression analyses they do with really large data sets of people in order to say, hey, methylation status of these things are strong predictors. These things are enabled by really important longitudinal studies like the Framingham Heart longitudinal study. This is something where they collect samples of people's tissue and DNA while they're alive throughout their entire lifespan, so that after they're dead, you can inquiry things about their biological status when they were 30, and then say, hey, at 30, this is something we can say about what happens at 70. So these are really important. And I think one thing that's relevant is that we know that methylation is important for the on-off status of cells, but in fact, there may be even something more associated with aging. So I want to talk about one particular theory of how aging actually works. Now, I mention this because this is the one that's most directly relevant to the clinical trial, the theoretical basis for why the clinical trial should work. It's not the only one. It's not necessarily the only one. These aren't necessarily mutually exclusive ways of thinking about how cells age. And Delia asked me that question at the end, because Delia asks about does knowing your methylation clock give you agency for doing something to help reverse it? And we'll come back to that. And so this is a nice pictorial depiction of what we think about as the chromatin, the on-off status of cells. And again, I'm going to use the term chromatin, and chromatin is a way to refer to this protein packaging of DNA being packaged in a way so some things are off and some things are on. And what you see here on the left-hand side is a nice orderly arrangement of chromosomes that are attached to the outside of the circle. The right genes are on, the right genes are off. This is a cell that is happy and, in a sense, young. But what can happen over time is that the methylation groups are not in the right places. The chromatin is not tight and well packaged. One way of thinking about this is like a closet. The left-hand side would be a nice orderly closet with nice little drawers, nicely folded socks. The right-hand side is disorderly. Things are strewn around. Socks aren't in pairs. Not everything is inside its own basket or on a shelf. They're just piled up. And once enough of that happens, it doesn't really function very well as a cell, just as a really disorderly closet doesn't function so great as a closet. And so this theory is one of the driving ways that people think about cellular aging and what's called the information theory of aging. Because remember, the on-off status of genes is a set of information that informs the cell what it should or should not be doing. Now, to go back in time a little bit, a lot of the outgrowth of this theory came from work in yeast by the Leonard Garante lab in the 1990s. And what I have here on the left-hand side is their assay for yeast, again, quote unquote aging, is that if you have a bunch of cells in culture and then you capture them with, they're constantly budding off a new, smaller version of themselves. But what happens is that every time they make a bud and then replicate, that actually leaves a little bit of quote unquote scar tissue. And over time, you can capture these cells out of culture and just count the number of scars. And so the more scars you have, the more you've replicated, quote unquote, the older of a cell you are. Now, there's another interesting aspect of yeast in that they have what are called mating types. So from our parlance, you might think of that as a gender, but they have an alpha gender and an agender. And the way they actually switch, like any given cell can switch that mating type by replacing the genetic content in a locus called the mat locus and then swap it back and forth. But what's really important, this is really important, is that the information that they're getting for the swapping, the original locations have to be silent. And so that's what you're seeing is that there are cassettes that are out there, but the HML and the HMR, which are the sources of genetic information, but they're not the gene, those need to be silent. And so when something is happening that they're becoming less silent, then you get different phenotypes you can measure. And that's actually what we see with older yeast, is that older yeast get kind of gender confused. And there are genes that are known to be involved with this. So these are known as SIR genes, the silent information regulators, right? Because these are genes that are silent, but they're not actually, and they need to be regulated in an important way. And so the gene SIR4 is something that has been shown to enhance longevity, right? So this is the idea of a genetic intervention. You can measure how old cells are in a population, and you can see a change in that. And the way these curves work is the number of generations is the x-axis, and then the number of viable cells is the y-axis. And so in this one particular mutant of SIR4, you see that with the filled in circles, that's shifted farther to the right compared to the other ones. And so these, this genetic variation is showing the shift in a population. And so this is something that's enhancing longevity. And what the Grante lab and other people have figured out over time is that these SIR genes are these silencing complexes. And what these silencing complexes are doing is helping maintain the silencing of silent genes, of what genes are supposed to be silent, but they actually sometimes have to move around the cell and then go back and reestablish the silencing. And so one way of thinking about, say, how one might measure the aging in this case, another way of actually doing it is that how much of this particular protein complex is where it's supposed to be to keep the silent genes silent. And so what I want to impart upon you is this idea is that there is this quantifiable thing, and where it's being localized and how genes are being silenced are all a part of an important aging process, is actually how we define and can see how this influences aging. Let's see. And so interestingly, these SIR genes, there are versions of them in mammals. Actually, there are versions of these in almost all the major model organisms that we study. They're throughout the kingdom of life. But these genes that are playing roles in helping localize silencing factors are present in humans and they're present in mice. And so back in the early 2000s, and this is a publication from 2006 showing the first knockouts of SIR-T6. So SIR-T is actually, they're also known as sirtuins, but they're called SIR-T because they're all orthologs or homologs of SIR2. And then of those homologs, they have their own numbering system in the mice. So there's 1 through 7. There's SIR-T1, 2, 3, 4, 5, 6, 7. Specifically SIR-T6, when you knock it out, these mice experience a highly accelerated aging. So you can see in the pictures of them, the ones that are KO, which is a term meaning knockout, meaning they don't have the gene anymore, they're frailer, they get gray hair, they're smaller, they're less energetic. Like I showed you before, the curve on the top right hand, if you're missing SIR-T, you see your mortality drop off very quickly, whereas the regular mice keep living on through the time span of that particular graph. Looking at the musculature and skeletal structures of these mice, you see they're highly compromised. And this is an important clue to what SIR-T6 might be doing, is that chromosomes start getting abnormalities. They start looking a little bit like cancer cells. And so the highlight here was, SIR-T6 may have something to do with one of the hallmarks of aging called genome instability. So this is something I hear almost every time I give an aging talk, and this is the power of the news media. Telomeres are associated in certain ways with aging, but more as a symptom of aging, not as a cause. Now, telomeres, these little caps at the end of chromosomes, these are something that are an ultimate limit on our mortality. But unless there's something happening first, they aren't really a cause of aging. And so that's, I think, something I want to say that's relevant to this talk, is that this idea, this information theory of aging, is actually not in any way about telomeres. And so further investigation in terms of this idea that SIR-T6 may be important for genetic instability came out later in research was done in the late 2010s, showing that if you do something to make a double strand break in DNA in a cell, so again, that's an initiating agent of DNA damage, that SIR-T6 shows up there. And it's showing up co-localized with a very well-established, well-known protein known as H2AX. And so that's what you're seeing in this picture here, is that the co-localization of that bright green spot with the dark red spot is saying that, hey, SIR-T6 is actually maybe something involved in double strand break repair and recognizing it. Yeah, so for those of you who are familiar with high energy physics or things like nuclear blasts, double strand breaks are a very immediate consequence of high energy radiation like X-rays, gamma rays, and in fact, one of the reasons they want you to wear a lead shield when you're getting an X-ray, especially on your gametes, is that those double strand breaks can be highly mutagenic and problematic for DNA. And so one of the ways of trying to study and recreate this natural phenomenon of double-strand breaks and genetic instability is to actually just create a molecular system that creates them artificially and then study what happens in mice that you've engineered to do this. So this is something the David Sinclair lab came up with. And what I'm showing on the left-hand side, and the main thing you need to look at are just two main things, is that there's a protein called PPO1, and when that is present and expressed, it'll look at double-strand DNA and make a break in certain sequences of it. Okay, it doesn't just go around breaking DNA all over the place. It breaks it very specifically, on average in mice, in about 10 to 12 spots, and makes a double-strand break. And then the other thing that happens is that the ability of this protein to be expressed is whether the mice are being fed tamoxifen or not. So tamoxifen is something that turns on this artificial gene, and then taking away the tamoxifen means that the cells are basically normal. And so the terminology for this system is introduction of chromosomal breaks. They call it ICE, ICE. And then CRE is just a negative control. CRE means that you're not actually having ICE, this PPO being expressed. And so what they're showing here on the right-hand side is that you expose mice to tamoxifen, they're under this ICE state, and suddenly they start aging at an accelerated rate. And there are lots of things that we can talk about, lots of things you can measure in mice, looking at their frailty, what other cancer predispositions they have, other things that happen. But there are two main things that are really important. And that is one, and that's here in this next slide, showing that as you induce this treatment, then you get a burst of stuff that's happening, and you actually see all this changes in the epigenome. In other words, the methylation status of all these cells are going into an accelerated aging type of state. They are getting to become like messy closets, and the cells are not necessarily understanding what it is they need to be doing. They've lost their identity, they've lost their program. The other thing that's really important here is that they're showing that the mutational burden, again, changes to the DNA after the breaks are made and then repaired, is extremely minimal. And this is really important because many models of aging, this accumulation of damage model, has always been the idea that mutations are occurring, and that's what causes aging. And while that can be associated, that can help cause aging, that's not necessarily the main driver of cells becoming older and losing this information identity. And so that's, I think, an important part of this. And that's what they're showing on the right-hand side, this little graph where you have a very short blue cloud, then a very tall red cloud, is showing that when you expose mice to all this extra double-strand breakage, they are increasing the amount of their epigenetic age because of this loss of a proper methylation status. So I hope that that's kind of clear what the system is, but the main idea is that messing up the epigenome, messing up the methylation status is basically a driver of aging. And what I'm going to do now is slightly shift gears to a topic I'm sure you've heard about, and it may not seem related, but we'll come back to it. And that is Dolly the sheep. So back in 1996, and I still, I put the slide together and said, my God, how was that 1996? Seems so long ago. But what's really relevant here is the idea that if you take an adult skin cell, right? So there was a donor mouse, donor sheep, that's what you see on the left. They took cells from it, cultured it in a dish, and then extracted just the DNA, right? So that DNA is what specified that particular sheep, that particular individual. And what they did with that DNA was from an ewe, they took out the egg cells, again, the eggs that are in the ovaries, which again, remember, these are much bigger cells. These are egg cells that are designed to help make an entire new individual once it gets fertilized. So these are bigger cells. They have the program of basically becoming every cell within the body. And so the idea here was, well, what happens if we take the nucleus of an adult cell and put it into this cytoplasmic soup of a cell that can become anything? And again, the idea here is something called pluripotency. Pluri meaning many, and potency is the ability. So pluripotency, the ability to become any cell. Although technically it should be totipotency, but we'll leave it at pluripotency. And so when they did this experiment and then implanted this egg with an adult nucleus into sheep, they actually, 1 out of, I think, 189 actually gave, or 3 out of 189 resulted in live births. And then 1, that live birth became Dolly, I think was the only adult out of that particular experiment. So one thing to point out here is that this is not an efficient way of doing this, right? This is not something where anybody, as rich as they may be, can turn around and say, I'm going to take my adult stem cells or my adult skin cells and make a new Stephen. That's an incredibly complicated and would be quite an expensive endeavor. But this idea, this core concept of reprogrammability has a lot of potential for what you can do with it, especially medically. And so there are 2 people to really credit here, and this was the Nobel Prize in Medicine or Physiology in 2012, awarded to Sir John B. Gurdon and Shinya Yamanaka. So Gurdon is someone who'd been working, I think, in the 70s, showing that this process that happened with Dolly is something he'd been doing with frogs since the 70s, and that there's lots of clonability of frogs. Take an intestinal cell, put it into an egg, you actually can get a clone of that original adult. And so, and this was very useful for frog study, because now you have a genetic clone where, for the most part, you can say we have a very similar genetic background. Although, again, not every phenotype and every feature is going to be exactly the same, because lots of things are dependent upon environment. Now, what Shinya Yamanaka was famous for was actually isolating the particular 4 genes, which are now called Yamanaka factors, OCT4, SOX2, KLF4, and c-MYC, where if you just take those 4 genes, express them at a high level into, say, skin cells, you can create induced pluripotent stem cells. And that was his award for the Nobel Prize. And again, this is something that from a medical point of view, you can think of lots of applications where, if we want to study a disease, we can recreate the organs or the tissues that actually that are affected by that. Or you can even create like replacement parts, or you can get some stem cells embedded in a person to say, and I think the most exciting option people were excited about was, say, a cervical spinal fracture, and that where someone is left paralyzed, you can find ways to regrow nerve cells or even cardiac cells. Cardiac cells do not divide anymore. You have a set number of those in your life as well. And so this idea of stem cells was very exciting in terms of medical applications. But, and this is where we bring the story back together, is that when we think about induced pluripotency, clearly you're resetting the cell back to a state of youthfulness, right? That's kind of the normal progression, is that you have a stem cell and then it differentiates. Is this something that also could reprogram the epigenetics of cells in a more gentle fashion to just de-age them? And so this is the work that David Sinclair's lab was working on, is the idea that if you take 3 of these 4, so again, don't include c-MYC, which is a well-known oncogene, and you express these at a high level in mammalian cells, either within tissue culture or within variations of mice, or you inject viruses that express these genes into mouse tissues, does this reverse aging? And so that's what I'm showing here on the left is this adenoviral technology of injecting it and then turning the genes on. Or on the right-hand side, they made whole mice that have this genetic program of expressing these at a high level. And so what's happening here is that these 3 of the 4 Yamanaka factors do accomplish demethylation without creating cancer. Again, one of the really important things that are relevant here to this is that when they study these over the long term, without c-Myc being present, you're not creating cancer in these organisms. And so this is the data from the 2023 paper showing that if you look at these mice that in this case are now being induced with extra double-strand breaks, although again, this was based on the prior work that they had just shown before, is that in mice where you've induced these double-strand breaks and messed up their epigenetic programming, their methylation status, if you go back and re-add OSK, you actually put them back to a younger state. You start reorganizing their methylation status. And that's what you see here on the right-hand chart, is that Cree mice, again, these are ones that are just kind of normal wild-type mice. That's the way their methylation status looks. The higher is the higher it is, the better. That's the proper methylation status. If you add all these double-strand breaks, you get the red in the middle, where now you have methylation not being where it's supposed to be. You get a little bit less. But then if you add back the Yamanaka factors, the OSK, and you get the green cloud, you're seeing the green cloud looks a little bit more like regular mice, as in the Cree. And so what's happening is that these three factors, which are things that tell genes whether to be on or off, but are really, in a certain sense, a button that says you are more like a stem cell than a differentiated cell, then it actually has this effect of de-differentiating them. So let's think about this in a more specific, particular tissue type of examination. This is what David Sinclair's group did, was that they were looking at blindness. Now, one thing to keep in mind is that your optic nerve, which is where a lot of blindness issues are. Again, this is not like, other types of things that can happen in terms of blindness, but this is very specifically things related to the optic nerve, where there are ganglion cells that are really important for replenishing and healing wounds. And this is another really important thing, that in mice and in humans, we have these ganglion stem cells in our optic nerves that can replenish tissue when we're young. So shortly after birth, I think by a year after birth, human stem cells can't replenish damaged or broken cells. But we did at one point have neuronal stem cells in these tissues in order to replenish tissue. And so they did the same thing. They had this, again, instead of using, in this case, they're using tetracycline. Tetracycline is an antibiotic that perfuses the body and can be used as an on-off signal for genes. Is that within these mice, either with adenoviruses or the mice having them, is that they would take the optic nerve, make sure it had these extra Yamanaka factors, and then use tweezers to crush them. And that's what you're seeing in the images, is that where the little stars are, that's where they created the crushed optic nerve. And then they went back and said, let's turn on the OSK Yamanaka factors and see what happens. And what they're showing in the lower hand side in the images is that you're actually getting a lot of activity and growth and ganglion cells that are now going back and replenishing and healing the tissue. So what they're showing specifically in this one is just expression profiles of these genes, but what they examined within the optic nerves themselves is a replenishment of tissue and regaining sight. And so this again is a measurable way of looking at and having an intervention where resetting cells to a younger age and having them become stem cells again is a medical intervention. Delia is asking a question about other cells and stem cells. Again, I'm not a physiologist, and so what I will say is that different tissues maintain stem cells when they need to be highly replenished, right? So our intestines, our epithelium, we have lots of epithelial stem cells. We have lots of immune stem cells, but we don't have neuronal stem cells because when we, for whatever reason, we don't replenish brain cells that are neurons in and of themselves. Whereas peripheral nerves can regrow, but say our brain stem cells don't. So again, this is let's just keep a focus on this particular system because this ends up being highly relevant to human health conditions. And so David Sinclair is, I think, on the board, or is it, he started up this company called Life Biosciences. I don't think he's actually the CEO of it anymore, but I don't know offhand. But it's based on this idea that if you can reprogram cells. No, if you see David Sinclair, you look at him and say, like, this guy does not look 56 years old. And you can see lots of YouTube videos about a lot of the things that we're going to talk about in just a second, of daily supplements he takes that in theory are ways of maintaining people as youthful. But let's come back to that later. He himself is in his mid-50s, and he's a very vibrant guy, actually. And so, oh, sorry, okay, so Sid is just pointing out he does other things too. If you listen to him talk about what he eats like no purified sugar. He doesn't eat snacks. He also exercises. He maintains a lot of brain health and activity. So again, I should say there are lots of pharmacological things he takes, but he also has a lifestyle. Yeah, we can talk about the specifics a little bit later. All right, so but his company said, and has now announced that for the very first time anywhere in the world, that this idea of euthanizing or de-differentiating cells is a medical intervention. And what they're looking at are, again, taking OCT4, SOX2, and KLF4, OSK, and injecting them, like you saw in the mice, into the optic nerves of a variety of patients that have optic neuropathies. And so, again, we're talking about retinal ganglion cells. These are these stem cells that can replenish cells that, again, stop working as stem cells once we're about a year old. And so, just losing these, like an open angle glaucoma, in which there's some neuropathology of the stem cells. Again, glaucoma, while it's highly characterized by these increased ocular pressures, that's not always necessarily the main cause of the blindness. It's actually just associated with some things happening in the optic nerve. And there's also what's something called non-arteritic anterior ischemic optic neuropathy, or NION, or stroke of the eye, where again, you can imagine in any situation where you've had a sudden and even short-term loss of blood supply, cells die. And so these are things where there's no treatment, right? The optic nerve, like a lot of neurology problems, it's hard to treat these because you don't have a way to replenish the cells themselves. And so that's what they're doing. And again, what I have here is the clinical trials ID. So you can look this up on clinicaltrials.gov, where what's designed, it's phase 1. They're going to recruit patients that have these particular pathologies, deliver these to retinal cells, and make sure that it is safe, make sure that it is well tolerated, and then measure markers of how much it actually helps the condition. And I think actually that's another point too. And I didn't go back and double check all of the specifics, but in a lot of these trials that you have for things helping with blindness, you actually have two cohorts. You actually have a cohort of different people, so you can measure what's happening to them. But in these cases, you also only treat one of the two eyes. And that way, within each patient, you have a cohort of treated eye versus non-treated eye. And in fact, it's a very powerful way to get good statistical data to show that something is having an effect. So I would have to go back and double check whether that's truly the case, but I know that's how that's been done with some, say, for example, CRISPR treatments. So anyway, it has started up just recently. They're expecting the completion of the data collection, everything in 2027, but the actual study itself, and this could be a very long wait, is not until 2032. Again, they will publish results as they get stuff. That's pretty common in these, but again, the actual final trial date and data completion will be 2032. So, that's exciting. I mean, I hope I set up the story to tell you about the rationale for why that clinical trial makes sense, what they believe it'll work, the fact that there is a theory behind it that is related to aging, but in this case, very much related to tissue regeneration, and that there were very effective things shown to work in mice before we transitioned into humans. So there are other things going on, other clinical trials, and these are things that are supplements that or other drugs that have been shown to have an effect on people's metabolism and markers of aging. So again, a lot of these things are places where there have been research studies showing that you can have various positive effects related to aging, or people looking at methylation clocks. And so what I have listed on the left-hand side is a key that one of the core compounds around all this is something known as nicotinamide mononucleotide, which is something related to niacin, in other words, vitamin B3. But it is a really important energy molecule in the cell. And again, without going through it in a lot of detail, is that these supplements and how they're working may be related to two central ideas. And that is this energy metabolism is really important for how cells do DNA repair. And so that if you have plentiful and abundant energy metabolism, you have good DNA repair. And that some of these other things, like you might have heard of metformin or rapamycin, that these are things that affect energy metabolism. But one of the aspects of energy metabolism is that it frees up energy in order to do DNA repair. So again, without going into a lot of detail, but one of the best examples is calorie restriction. That if you restrict the amount of calories you're bringing into your body, that the ultimate physiology effect of that is that you actually have more free energy to accomplish good DNA repair. And so now David Sinclair, in his information theory of aging, would say, and his interpretation of all this would be, is that the main effect has to do with these silencing modulators, that what you're really improving is how those work, and that that's how you get the improved DNA repair, you maintain a methylation status that's proper, and that that's an aspect of it. So again, I'm not necessarily promoting strict calorie restriction as a health advice. I'm just saying from a scientific point of view, this is something people have been working on and showing works within these axes within model organisms. Again, one of the best ones is the nematode. The DF4 mutation is a very telling one having to do with glucose metabolism. So where are there clinical trials in the space? And so I won't talk too much. I won't get into the details, but reverse transcriptase inhibitors are something that may reduce the DNA damage burden in cells. And so there's a clinical trial called Line-AD to see if Alzheimer's disease can be somewhat alleviated or progress more slowly if you're limiting the amount of DNA damage that's happening in neurons. Senolytics. So I haven't talked too much about senolytics, but this idea of cells being healthy. And if cells get deprogrammed, one thing they become is called senescent. And so a senescent cell can also tell other cells become senescent, even if they're already healthy. And so a senolytic is something that kills senescent cells. And so this idea of senolytics is something very popular. And so there are interesting clinical trials having to do with this, where if you can remove senescent cells from your body, then on average, you have more healthy cells around. And that is also, again, associated with, again, as you would measure methylation, you would get rid of the bad cells in that case and have more plentiful fresh cells. The other question too is also if senescent cells get in the way of healthy cells too, so it can help alleviate that. And so they're looking specifically at osteoporosis. Metformin, which I just mentioned, may be involved in energy metabolism. We know it's a drug that's been used to treat diabetes for several decades. Is that they're looking at that as a way to maybe also help with cognitive decline. Again, you might have heard of something called type 3 diabetes, which we now think may be a key aspect of the symptomology of Alzheimer's disease. But metformin, again, is something also that you can think of as an anti-aging drug. Does it help with cognitive decline? And here one, to bring it all back to full circle, and this actually may be the one drug that'll be the big money maker of all the ones I've mentioned so far, is this CS002, which is also an mTOR inhibitor, but there have been small pilot studies showing that it helps treat gray hair. So again, coming back to what might end up being the most successful of these drugs, if it actually does pass clinical trials, might be the one that I think affects the least consequential part of the aging process. And so, again, this is just a highlight of some of these. If you were to look up any of these drugs I've mentioned, like metformin, senolytics, you can do searches for clinical trials. You'll see them all over the place. One thing I will say, as I was searching through some of these, I was looking at older trials where a lot of NMN trials or NR trials have not shown the efficacy that was expected out of them, which again doesn't necessarily say that the theory is not correct, but that they didn't have the power to affect things within a clinical population as you might have hoped. So in summary, I want to just remind people that methylation, again, this theory of aging is that methylation is a key element of cellular identity. And that's known, that's well established, and that may be a key relevant factor to how aging occurs within human cells. Erosion of this leads to dedifferentiation, but you can reestablish youthful states with interventions. Stem cell-like acquisition can heal tissues in situ. We definitely see that in mice, and now we're having a clinical trial to see if that can affect humans, which would be exciting. And that ultimately Ponce de Leon should have been looking in Boston. And that's where David Sinclair and Harvard and a lot of this relevant research has happened. Although, of course, there are researchers all around the world who are working on this and doing exciting stuff. So with that, I would like to thank you for your participation and attention for today. And I'm happy to take any questions, and I'll stick around for a little while. So feel free to point to ask your question local chat. Although the recording will probably end right about now. So thanks again. See you all next week.